Chapter 5, Chemicals and Cancer. In this chapter, we have two major themes. One is identifying chemicals that cause cancer, discussing some of the first known carcinogens that were discovered, some commonplace carcinogens, and also how do we kind of study that using animal studies and things like that. The second theme is mechanisms of chemical carcinogenesis. So we briefly mentioned the different categories of chemical carcinogens and then mentioned how they can be metabolized by the liver and how they can cause DNA mutations. And finally, we will talk about the idea that the development of cancer happens in a multi-step process. So we have tumor initiation, tumor promotion, and then tumor progression. So that's kind of the overall flow for this chapter. When we say the term carcinogen, this is meaning that this is some substance that's able to cause cancer in living tissue. Generally, we think of carcinogens as chemical carcinogens, but we also have physical ones and oncogenic viruses or cancer-causing viruses. So most carcinogens can fit into those three categories, and we'll give examples of each. Some well-known chemical carcinogens would include asbestos, benzene, radon. You've probably heard of those in your lifetime. And then just a note that carcinogens do not cause cancer at all times, under all circumstances. A lot of times it has a lot to do with the dose of which a person is exposed, the time of which they're exposed, and also just by chance if that carcinogen caused a mutation at the time of exposure. So thinking back through history, it's kind of interesting to think about what was the first carcinogen that was identified. And so this chapter kind of covers some of the history of the identification of known carcinogens. So looking back through history, it's interesting to think about the first identified carcinogens. And here we have a couple of examples. We have Dr. John Hill in 1761, who reported that people who use snub, which is a type of powdered tobacco, have a high incidence of nasal cancer. And he put together that there must be something in the tobacco that's causing cancer. So this is listed as our first identified carcinogen. Not too long after that, we have Dr. Percival Pott, who reported high rates of scrotal cancer in young chimney sweepers. And so he identified an environmental factor that he thought caused cancer, and he recommended them to use protective clothing. Some other examples we have are in the 1900s, skin cancer detected among workers of coal tar industry. And then in the 1890s, bladder cancer detected in workers of factories that produced aniline dyes. And so we are going to go into that example a little bit more. But these are some of the early examples of showing that that there could be a carcinogen that these people are exposed to that directly relates to them developing cancer. In the late 1800s, we have William Perkin, and he was trying to synthesize quinine, which is a drug to treat malaria. But in the process, he discovered mauveine, which is a synthetic dye that has the ability to stain things purple. So noticing that this is able to stain something purple, he opened up the idea that maybe I could use this as a dye in industry and stain, like fabrics and things like that with it. So large-scale production began of the chemical that's needed for making this type of dye. And the chemical needed to, as a starting material for this, was 2-naphthalene. And so now we have these factories, and we have factory workers who are being exposed to 2-naphthalene, and their goal is just to make this synthetic dye to make these beautiful purple fabrics. So very exciting for the ladies. Get to wear these new fabrics with this new dye. But then at the same time, we're exposing these factory workers to this new chemical that they hadn't previously been exposed to. Well, over time, it was noticed that many of these factory workers developed bladder cancer. And so looking back through time, it actually came to be known that nearly all of the workers exposed to 2-naphthalene experienced bladder cancer. And so we see here, what's listed here is that 15 out of 15 workers developed it. And that exposure was delayed. The development of cancer was delayed 10 to 30 years from their exposure. And then it took 50 total years for large-scale production to stop. So it took some time for, kind of the evidence to come out and for people to really believe that this chemical was causing cancer. So this is one of our first examples of like a large-scale carcinogen causing cancer and then having to do something about it. In this graph, we're looking at bladder cancer rates in people that are exposed to 2-naphthalene. And this is broken out into years after initial exposure. So 2 years, 3 to 4 years, and then 5 years. And what this graph is showing us is that if you have less time of exposure, you have lower bladder cancer rates. So here we have about 20% for people that have less than 2 years of cancer exposure, of naphthalene exposure. Here we have 3 to 4 years exposure, and we have a very high rate up here. We have somewhere between 60 and 80. And people that were exposed for greater than 5 years have almost 100% exposure. So very, very clear, very clear suggestion that these two things are linked. So maybe you can take a second and just think about what are some other carcinogens that you are familiar with. I know sometimes we hear about these terms in passing, and you may not know about them in depth. But maybe you can just take a second to think about what is something that I already know about. And we're going to give you an example of a very common one. So asbestos. Asbestos is a carcinogen. And the story about asbestos is interesting because it was very, very common in our environment for quite a while. It was used in roof shingles, ceiling, floor tiles, insulation on pipes. So if you were in a house or, your work office, this asbestos would be everywhere, right? And so for a long time, people didn't realize that it actually was a hazard. But come to find out that it was shown to be linked with the development of lung cancer and especially mesothelioma. And what has been discussed about asbestos is that it is released into the air as these teeny tiny little fibers. And it's the fibers, the shape of the fibers and the fact that they cause inflammation. When you breathe them in, it's almost like breathing in teeny tiny little pieces of glass or something. So it causes a lot of inflammation inside of your lungs, and then ultimately it leads to cancer. So thinking about things that we actually produce, we produce tobacco, which we know is a known carcinogen, and then asbestos would be the second most lethal product that we've produced. And what's terrible is that this is now in people's houses and in people's homes. It's not exactly easy to get rid of asbestos. But there was a link found between asbestos exposure and lung cancer, specifically mesothelioma, which is a rare form of cancer, prior to us using asbestos in our buildings and things like that. So in this graph, we're looking at years, so 1970 to 2000, and annual deaths from mesothelioma. And we see that the rates are increasing. So the ban on using asbestos was, I think, about the late 1980s or 1990s. So if the ban occurs somewhere around here, then why do you suspect that you still see this increase here? So you might be remembering that there is going to be some time delay between the exposure to the carcinogen and then the development of cancer. It could be 10 to 30 years delay. So even if asbestos was removed at this point, people are still going to be presenting with their disease later and later after that. Here we have a list of carcinogens and cancers associated with occupational exposure. And you can find lists of carcinogens on the U.S. National Toxicology Program website. And so they keep a list, and obviously they keep tabs on all of this. So they consider the testing that's done on these various chemicals and determine whether it is a true carcinogen. And then they also link it together with the type of cancer that they're known to cause. So obviously, before a chemical or substance is listed as a carcinogen, it has to undergo rigorous testing, and oftentimes this occurs in animal species. So once we have this running list of carcinogens, the next kind of question is, well, who is going to regulate that, right? Someone has to oversee that these factories or industries are not using carcinogens and exposing factory workers or people to these chemicals and causing a higher risk of cancer. So something has to oversee this whole process. So we do have a government agency that is responsible for protecting the safety of workers. You may have heard of it before. It's called OSHA, Occupational Safety and Health Administration. So this is a government agency that is in charge with making and also making sure people follow regulations that protect the safety and health of workers. So their goal is to eliminate dangerous chemicals or to at least limit the worker exposure to below a dose range that would be known to cause cancer. So due to the work of OSHA, there has been a decrease in occupationally induced cancers, down to 5% of all cancer deaths. So we've been talking about factories, industries, things that we use for building. Another thought when we're thinking about carcinogens is, okay, I'm thinking about factories, I'm thinking about people making various things. What about industrial pollution? What about during the process of which they're making their final product? Some of these chemicals are being released out into the air. So are those chemicals responsible for increased cancer risk and death rates? So as time goes on, obviously we're making more factories, we're making more end products, and we suspect that we're also adding to pollution in the air, right? So in this graph, we're looking at the year, so time, 1930 over to the 2000s, and we're looking at the production rate for pesticides and plastics. And so we've got those graphed here on the graph. We have pesticides increasing rapidly, plastics increasing very rapidly. And then looking overall at breast cancer and colon cancer rates over this time. So what we see in this graph is that breast cancer rates, sometimes they rise and lower a little bit, and here they lower after around the 1990s due to improved cancer treatments and also detection measures. Here we have colon cancer, and we see a slow, steady decline over time. So what this graph is suggesting is that there is not a clear link between increased industrial pollution due to the production of pesticides and plastics on the rates of breast cancer and colon cancer. When talking about chemical carcinogens, we can discuss the principles of chemically induced carcinogenesis. Number one would be long time delay, which we've talked about, is that between the time of exposure to the time of development of cancer, it could be up to 30 years. So there's a long time between the event of exposure and the development of cancer. Number two is dose dependency. So if someone is exposed to a high dose for a long time, then that results in the highest risk of cancer. The lowest risk, of course, would be a low dose for a small amount of time. Organ specificity. So we've kind of looked and seen that some carcinogens preferentially develop cancer in certain areas of the body. And this all depends on the way that these chemicals accumulate in our body tissues. It's possible that they might concentrate in the urine, and then be related to like bladder cancer, for example. Now, kind of going back to our previous topic a little bit. So, thinking about that pollution out in the air. So, we kind of just usually have a general feel that, okay, if I live next to these factories and they're shooting these chemicals into the air, that's got to be bad for me. That's got to increase my rate of cancer, right? But I kind of feel like, oh, I've got my house, the air inside of my house has got to be safe. You know, I'm not making those chemicals in here. So, thinking about that question, do you think the air inside your home is safer than the air outside? In these graphs, we're looking at various chemicals that have been tested both indoors and outdoors. So we've got the orange bars are for indoors and the blue bars are for outdoors. Here we've got the different chemicals listed here, and these are all falling under the category of toxic, volatile, organic chemicals. And here they're being studied inside versus outside. So it's interesting, as you will notice, that in every case here, the indoor bar, the orange bar, is larger than the outdoor blue bar in every single case. And we're even talking about things like benzene, which we know is very dangerous for us, right? So this study is kind of proving, actually, indoors, we are exposed to even higher rate of some of these chemicals than we would be exposed if we were to go outside. So obviously that's going to create cancer risk, right? Here on the right-hand side, we have pesticides. So a lot of people worry about using pesticides inside of their home. So here are some of the chemicals involved in pesticides. And we see that their levels are much higher inside of our house versus outside of our house. So when we think about being exposed to chemicals via air pollution, it's interesting to think that we are actually exposed to more chemicals, in some cases, inside of our house than if we were to go outside. So a few of the reasons that happens is because we use chemical compounds to clean our house. We use chemical compounds for painting. We have chemicals in our carpets, our air fresheners, our disinfectants, and all of these things are contributing to the amount of these chemicals that are in our air inside of our house. The doses are still really, really low and much lower than they would be if you were in an industrial workplace. So it is a little hard to assess the risk of these low dose exposures, but it's kind of interesting that the exposure we're getting is actually higher inside of our house versus outside. So we mentioned we are exposed to low dose of chemicals constantly. And so how do we understand what that low dose exposure is doing to our cancer risk? When we talk about epidemiological studies, they oftentimes are not sensitive enough to determine what our cancer risk is to these carcinogens when we are exposed at a very, very small dose. And so, as we mentioned previously, that a lot of times we want to study these chemicals in our animal models so that we can figure out what our cancer risk is. But the problem we mentioned before was that in order to do these studies, these animals had to be exposed to a high dose, and sometimes 10 or 100 times higher than what humans would be exposed to in the natural environment. So these studies are done using these very high doses, and then we extrapolate the data to determine what our risk is at the low dose. Many of these studies are designed to determine what the maximum tolerated dose is, MTD. And that term means the dose that is the highest at which you can give to that animal species without threatening their life. So there are experiments that increase the dose of the drug until they recognize that the highest dose of which the animal model is not able to stay alive. And so that's the MTD. And then sometimes future experiments are done using that MTD dose. And so if we're trying to extrapolate the data from animal models that have used that high dose, it's hard to say that we're really seeing the same thing, because an animal exposed to a very, very high dose of chemical is going to have a much higher cancer risk than obviously someone that's exposed at a very low dose. Looking at these studies, we also talk about the threshold. The threshold means that this is the dose that must be exceeded before cancer rates begin to rise, which is kind of mentioned here on this graph. We also have two major models. We have the linear model, which we mentioned before, that we have animals that are treated at a very high dose, and then because these doses are increasing in a linear way, then we can see the linear dose response relationship, and then we can extrapolate the data down to the lower dose that humans might be exposed to. If we look at the threshold model, we are able to look at a larger range, and then we can kind of use a different way to model that data. And here we see kind of like a sigmoidal curve. It's no longer a linear curve, but this kind of takes into account that at the lower doses we have a low rate of cancer, and at the high doses we have a higher rate of cancer. And then it also takes into account a threshold. So after this threshold, if you increase the dose beyond the threshold, there is a cancer rate. If your dose is before the threshold, then there is zero cancer rate. So looking at our various models for how we can study the relationship between carcinogen dose and cancer risk. So number one, we have the linear model, which assumes the linear dose response relationship with no threshold. So again, you're just using increasing doses of drug and assessing the cancer rate, and you're assuming it's going to have a linear relationship. The threshold model involves a threshold dose, and it assumes that there's no cancer risk at the lower doses, followed by a relationship between cancer risk and the higher dose of the drug. In some cases, this can overestimate human cancer risk just based off of that modeling system. But over here, in some cases, it can also have kind of like a bad extrapolation to say that something has an effect at the low doses, but maybe it doesn't. Finally, we have the hormetic model, which involves the threshold dose, and it has the U-shape. So this takes into account that high dose of the drug is correlated with higher risk, and that low dose of the drug is correlated with no or actually a reduced rate of risk. And when we look at this dashed line, we're looking at the background cancer rate in absence of the carcinogen. So what is the normal cancer rate? So this is saying that this is actually below it at some low dose, and at some dose it's higher. So that's an interesting concept, thinking about, could a lower concentration of a chemical actually lower your rate of cancer, but then a higher dose of it could increase the rate of cancer? So thinking about this, think about a chemical that if you are exposed to this chemical, it could cause some kind of tissue destruction. It could cause some kind of problem with your cell. Your cell recognizes that something is wrong. So in this case, it could activate a repair mechanism. And that repair mechanism could fix errors that are occurring in DNA and could prevent cancer from arising. But then if you go to a higher dose, that you're no longer in this kind of protective repair phase. You're actually now in like major DNA damage, major tissue damage, causing a lot of problems kind of phase. So that's what this model is taking into account. And you'll have to excuse me because I made an error. This should say low dose exposure instead of high. So to kind of summarize, there were some limits to deciding if low doses of carcinogens correlate with cancer risk. We mentioned that epidemiology, those methods were not sensitive enough to detect small increases in cancer incidence. And so we must use animal studies. And animal studies do a good job, but then we have some problems as well. Because we can't necessarily use that low dose either in the animal models. So we have to use a higher dose and then extrapolate our data to see what we think the risk would be at a lower dose. So one thing that is done is use the maximum tolerated dose in these studies. And so this could lead to an overestimation of what the effect of a low dose would be on the cells. If we're using this high dose, then we're causing tissue destruction and cell death. But if we're trying to extrapolate what that means for a lower dose, we're not taking into account that that low dose is not causing tissue destruction and cell death, and maybe even for some cells, it's causing a protective situation and actually lowering the risk of cancer. So it's tricky trying to find out what the low dose of carcinogen is doing with our cancer risk. And we have some examples where sometimes the animal model doesn't give us perfectly relatable data. So in the first example, we have AAF, which can cause cancer in rats, but it does not cause cancer in guinea pigs. The reason why would be rats express an enzyme that makes the compound metabolically active. So there's a chemical reaction that happens that causes the chemical to become a true carcinogen. And guinea pigs do not express that enzyme, so they do not experience that chemical as a carcinogen. In the second example, we're looking at the artificial sweetener saccharin. So it was shown that they cause bladder cancer in rats. So then everyone was concerned, okay, if we're taking in all this artificial sweetener, are we also going to have a higher rate of bladder cancer? But then it was found that there was a correlation between rats and the fact that they ingest lots of sodium salt, including sodium saccharin, and that causes crystalline precipitates in their bladder, and those crystalline precipitates actually increase their risk for cancer. Now, if you're going to compare that to humans, we do not ingest that sodium. We do not get those crystals in our bladder, and so we actually have a lower risk of getting bladder cancer. So looking at these chemicals in different animal species could also be interesting and highlight the differences between the animal models so that we truly know what the effect is in humans. So the U.S. government does publish a list that lists all the carcinogens, and it lists them into two categories. One is known to be human carcinogens, so there's about 50 that are on this list. And the second list is reasonably anticipated to be human carcinogens, which the numbers could be in the hundreds. We've been talking mostly about chemicals, but there are some medications and some hormones that are known to cause cancer. And so they're kind of listed here with what they're linked to, the type of cancer that they're linked to. And this is, of course, going to be dependent on the dose that's given and the amount of time that the person is being exposed to this. So when we think about carcinogens altogether, we must remember that we had been talking about chemical carcinogens, there are physical carcinogens, and then there's also the oncogenic viruses. But if we look at chemical carcinogens, they can be kind of grouped into some major groups. So we have the polycyclic aromatic hydrocarbons, aromatic amines, and that you can see that they contain the amino group on them. In nitroso compounds, so it has this N double bond oxygen group. Alkylating agents, these are known to cause reactions with carbon-containing chemical groups. Natural products, so it's kind of surprising sometimes to see that there are natural products that are known to cause cancer, but aflatoxin is a well-known one of those. We have inorganic substances as well, so we had asbestos was our example that we talked about. So lots of different chemicals can fit into these various categories, and so this part of the chapter summed up how do we get about knowing that it truly is a carcinogen. We mentioned it's a big deal to be called a carcinogen, and that it's kind of interesting to note that while we think that there's so many chemicals that would be a carcinogen, that only 50 are on the list. So obviously it's kind of a big deal to get on that list, right? So you have to have all the evidence pointing in that direction. You have to have the animal models done appropriately so that we can truly understand what the risk is of developing cancer when exposed to those carcinogens. And we must also know what dose is the appropriate dose to stay under so that we can make regulations that are carried out through OSHA, and they handle knowing what is the maximum dose and what is the maximum time of exposure that workers should be exposed to, so that they minimize their cancer risks. So we will pause there. I'll do the second video on the second half of this chapter. Thank you.